Okay, if we scroll down, the next topic that we have is signal conduction in nerve fibers. We said we'll have two main types of fibers. We said we are going to have the myelinated and unmyelinated fibers. Okay, if we scroll down, we can see here this unmyelinated. You see there's no myelin sheath covering the axon. And scroll down, and we can see the other one where you have right here the axon, but it's covered with myelin sheath. So the main characteristic of these fibers is that one of them is going to be faster than the other one. And for that, we can fairly explain it with this. We can say this one, remember, inside is negative. It needs to become positive, right? Sodium goes inside, so everything goes from negative to positive. In order to do that, you need to open channels for sodium, and that allows sodium to go inside. So let's say that from here to here, there are 100 channels for sodium. So, and you need 1 second to open each channel. Okay, just for educational purposes, let's say 1 second. So then how many seconds it will take you to take the information from here to here? Since you have 100 channels, then it will take you 100 seconds because you need to open every single channel from this point until the end. Okay, so then how long does it take you then to go from this point to this point right here? It's going to take you 100 seconds. So let's scroll down. Let's take a look now at the myelinated fibers. Myelinated fibers right there, the same concept. You have right here the axon, okay, and we have 100 channels, same as the one before. So we said 100 channels for sodium. Cell is, the neuron is negative, okay. Now channel for sodium will open and sodium will go inside. However, you can see here that yes, we have 100 channels from here to here, but most of the channels are going to be covered with the myelin sheath, which is this. So the only channels that are going to be open are only these channels that you find in the spaces in the nodes of Ranvier, which are the spaces between each set of myelin sheath. So you see that's why in here you see negative, and you see positive, and you see positive only in the spaces. So if we said that we have 100 channels from here to here, then you don't need to open all the 100 channels because most of the channels are actually covered with myelin sheath. So then, let's say that in here, then how many channels we have? Let's say we have 2, 4, 6, 8, 10 channels. So then, in this case, of the myelinated fibers, we need to open 10 channels only. Since we said that we need to use 1 second to open each channel, so in order to open these channels and in order to allow the transmission of the impulse in the myelinated fiber is going to take you only 10 seconds. Why? Because you need to open only 10 channels. And as you can understand, 10 seconds is faster than 100 seconds, which is the one that we were talking about right here with the unmyelinated fiber. So this one is going to take you 100 seconds. It's going to be slower. In the other case, it's going to be 10 seconds, therefore it's going to be faster. So in conclusion, the myelinated fiber transfers the impulse way faster than the unmyelinated fiber. Since in this case you need to open every single channel, then this is called, in the unmyelinated fiber, is called continuous transmission. But in the case of the myelinated fiber, like in this case, since you need to open only the ones that channels that you have in the nodes of Ranvier, it jumps from here to here. The stimulus jumps from here to here. The stimulus jumps from here to here. So this one is actually called saltatory conduction. So in the myelinated fiber, it's called saltatory conduction. In the unmyelinated fiber, it's called saltatory conduction. Right here, for example, you see saltatory conduction. Okay. So then let's keep scrolling down, and that's about it for this part. Okay, the next topic is synapses. Synapses are the area where two nerves get together in order to transfer the information from one nerve to another. Similar to what the neuromuscular junction is, but in the case of the neuromuscular junction is a communication between a nerve and a muscle. In the case of a synapse is a communication between a nerve and another nerve. Okay, so we can scroll down and we can see here, for example, you see, you have here in brown one nerve and you have lots and lots of other areas, right, of the other nerves getting in contact with it, right? Remember, this is the it's going to be the synapse, and in the space in between these areas is going to be the synaptic cleft. One thing to understand using this figure is that that nerves don't connect only one with one, right? We don't have only two nerves. We have lots and lots of nerves, and as you can see here, lots and lots of neurons, and they connect to lots and lots of other neurons. Okay, so the concept in here is, what type of stimulus is this? Well, you have to understand we have two types of stimulus. One of them is positive, and the other one is going to be negative, or excitatory and inhibitory. For example, if you move your hand, you're not going to move your hand forever, right? You need to stop that movement. So in that case, the movement of the hand is going to be excitatory. The stopping of the movement of your hand is going to be inhibitory. Okay, so that's a very important concept. It's like in a car. In a car, the car runs because you have an engine and you have gas. But that car doesn't stop just because you remove your, let's say, shoe from the gas, right? You actually need to hit the brakes in order to stop the movement of your car. So you have two systems. One of them, let's say, is gas, and the other one is brakes. Same thing in here. So what do you think this neuron in brown is going to do? Well, it depends. If, let's say, we have in here 100 other neurons that are getting in contact with this through the synaptic cleft, then the response is going to be either positive or negative, excitatory or inhibitory, depending on maybe 70 of these ones are going to be excitatory and 30 of them are going to be inhibitory. Then the result is going to be excitatory. Why? Because most of them are going to excite, most of them are going to be positive, most of them are going to make the car run. And because of that, this is going to be, for example, excitatory. The same thing can happen the other way around. If, let's say, 80 of these are negative and only 20 are positive, then what this other neuron in brown is going to do is going to stop. In this case of the example of the car, you're going to hit the brakes. Okay, so then you have to understand that you have excitatory and you have inhibitory. And it's not only one-to-one, but it's a whole bunch of them trying to get the answer, or trying to get a response from the other neurons. When we talk about the neuromuscular junction, we said that the nerve is going to release acetylcholine. And acetylcholine is going to make you move your muscle. Okay, so then we can consider that the neurotransmitter acetylcholine is excitatory because it's going to make you move your hand. But you have to understand that it cannot be that the same acetylcholine is going to make you stop your hand moving, right? So that means that there are different kinds of neurotransmitters. And that's the next topic that we have in here, neurotransmitters. Obviously, we are not going to talk about how the neurotransmitters were discovered or anything like that, but there's something that you need to know, which is the structure of how these neurotransmitters are going to be. Okay, what are they going to be formed? You have an example in here of how the neurotransmitters work, and this is obviously something we already covered with the neuromuscular junction. The nerve releases a neurotransmitter, and in this case, since this is a nervous system, this is another nerve which has receptors for the neurotransmitter. Okay, I think that's something that we already covered. And right here you have the neurotransmitters. Okay, there are different kinds. Some of them are going to be excitatory, some of them are going to be inhibitory, the same way the gas makes your car move and brake fluid makes your car stop. But that's basically what you need to know about the neurotransmitters in our case for this class. There you need to know that there are different kinds and they fall under the category of acetylcholine, amino acids, monoamines, or neuropeptides. That's all you need to know, that there are 4 main groups, and these are the main groups that you have to know for this class. Obviously, within each class, you're going to have excitatory and inhibitory, and if you scroll down, you see you have a table in there that is going to show you the different kinds of neurotransmitters that we have fall under each category. Okay, so if we scroll down, for example, some of GABA, for example, is inhibitory, as we were saying, so that's going to make you stop. And we said acetylcholine is excitatory. Okay, synaptic transmission. They're just giving you examples in here. We said excitatory has to be acetylcholine, right? We already mentioned that. So acetylcholine is going to help you stimulate that. It's going to help you move your hand, for example. Inhibitory, I told you that inhibitory can be GABA. That's one example that you have right there, right? In this case, that should be like the brakes. So if we keep scrolling down, you have more examples. And cessation of signal. So how do you stop these signals from continuing transmitting? Then you're going to take back the neurotransmitter into the neuron. That's reuptake. In some cases, the neurotransmitters get released, but they reach areas where there are no receptors. So they just get, they diffuse to these areas and then they get reabsorbed and destroyed. And the other one is degradation at the level of the synaptic cleft. Like in the case of the acetylcholine, there's an enzyme in there called acetylcholinesterase that is going to destroy that particular neurotransmitter. Another thing that you need to know is neuromodulator. Neuromodulator, they enhance the response achieved by the neurotransmitter. Okay, some of the most common neuromodulators are going to be, for example, nitric oxide. So the job is just to enhance the response of the neurotransmitter. Okay, and let's see, let's keep scrolling down. And that's it for this chapter. For this part. Okay, the next topic is neural integration, which means that you have a lot of neurons and they work together, right? But each group of neurons, even though they work together, everything is coordinated. For example, you have neurons that help you with vision and you have neurons that help you with hearing, but you can do both at the same time, right? And remember, I showed you the previous figure where you have one neuron and lots and lots of other neurons attaching to it, providing you with a response, which is that figure right there. Okay, I just went back a few pages to show you that. And I said if most of them are positive or excitatory, then you're gonna get an excitatory response, like you have it in here. And if most of them, let's say 80 out of those 100 are negative, then you're gonna get an inhibitory response, as you can see right there, inhibitory response. Because we said that they can be excitatory or they can be also inhibitory, right? Okay, so we keep scrolling down. Excitatory, inhibitory. We said summation, facilitation, and inhibition. That's the same thing that we talk about. If you have 80 that are going to be positive, one of them is going to give you one stimulus. The second one, another stimulus. The third one, another stimulus. The fourth one, another stimulus. So little by little, you start adding up, right? That's summation. Okay? So then inhibition is the same process. And facilitation is the same concept. So you just continue stimulating, whether that's going to be excitatory or inhibitory. So in here, they are trying to show you examples of summation. Remember, I showed you that figure where you have a lot of them and they can produce just a few of them, these stimulus, to get the response, or maybe one of them is just enough to produce the response at the level of the other neuron. But again, it's the same concept. You need to add them up in order to get the response, because a lot of times one of them is not enough. Same concept in here. Excitatory, you get 1, 2, 3, 4, 5. Eventually, you're going to get enough in order to get an action potential. Okay, I think that's fairly simple. We already explained that. We keep scrolling down. Same thing, inhibitory and neural coding. This is what I was telling you, that groups of nerves are going to be specific for vision. Some other nerves are going to be specific for hearing, but they are already coded that way. They are already labeled that way, especially for quality, meaning, for example, the eyes make you see light. So even though you are touching your eyeball, for example, with your finger, touching is not light. However, the eyes respond by producing light when you touch your eyes. Okay, that's quality. And quantity a lot of times depends on how many neurons are going to fire. So for example, in here, if it's only 2 grams, only a few neurons fire. But if it's 20 grams, a lot of neurons are going to fire. So it's give you an idea of how strong the stimulus is. We keep scrolling down. The next topic is neuronal pools, because it's a pool because the neurons don't work alone. They work in groups. If we scroll down, we are going to get to this figure where we have the neuronal pools. Okay, so the first one is divergence. So for example, you have light, goes to this set of neurons, and with light, you will be able to see with this set of neurons. You're going to be able to maintain equilibrium with this set of neurons, and this set of neurons are going to tell you that you are awake. Converging, for example, this is the nerves that are going to help you move your diaphragm. When you inhale, these nerves are going to help you inhale and you move your diaphragm. But in order to exhale, you use these other nerves and that's going to help you move your diaphragm. Reverberating, using the same example of inhaling and exhaling. This one is inhale, exhale, inhale, exhale. You need to go back in order to trigger this process again. So that's what is called reverberating. Parallel after discharge, you step on a piece of glass, you say ouch with this one, and you move your feet away from the piece of glass. These two things happen at the same time, and that's reverberating. That's parallel after discharge. Okay, scrolling down, we can see here the definition of parallel after discharge, but the next topic is memory. Okay, so let's take a look at that. There are 3 types of memories: immediate memory, short-term memory, and long-term memory. Immediate memory is the one that you remember only for a few seconds. Like, for example, you are reading something and you remember just those words.